This is the second and final presentation about the chemical level of organization, Chapter 2. In this lecture, we will cover the four major organic molecules, which are carbohydrates, lipids, proteins, and nucleic acids. We will also define ATP and its role in the cell. To begin, let's get some background on organic molecules. Organic molecules always contain carbon, and they are a good source of energy. Organic molecules do not dissolve easily in water, and they are arranged with a carbon skeleton and a functional group that's responsible for the molecule's characteristic chemical properties. When we discuss organic compounds, it's important to use proper terminology. A monomer is a small molecule that acts like a building block. When several monomers join, often by dehydration synthesis, a large polymer is created. So a polymer is made of many monomers. Now let's go through four important organic molecules. The first one we'll discuss is carbohydrates. Carbohydrates, or carbs, are sugars and starches. They are made of carbon, hydrogen, and oxygen. Carbohydrates function in making ATP, the energy used in our bodies. Carbs are found in three forms: monosaccharides, disaccharides, and polysaccharides. Monosaccharide means one sugar. Monosaccharides are the monomer for disaccharides and polysaccharides. Here we see the comparison of the three types of carbohydrates. Starting on the left with monosaccharides, these are simple one-sugar molecules and are the monomers of the more complex di- and polysaccharides. Examples of monosaccharides are glucose, fructose, galactose, ribose, and deoxyribose. When two monosaccharides are combined, a disaccharide results. Sucrose is a disaccharide made from glucose and fructose, two monosaccharides. Lactose is another disaccharide. It's made from galactose and glucose bonding. And maltose is a disaccharide made from two glucose molecules combining. Polysaccharides are large molecules made of many monosaccharides. These are important because this is how excess sugar is stored. When there's a surplus of sugar, polysaccharides are created. And when their sugar is in low supply, the organism breaks down the polysaccharide into smaller sugars to make ATP. Animals, like us, store excess sugar as glycogen, while plants store theirs as starch. The second group of organic molecules we'll discuss are lipids. Lipids are commonly known as fats and oils. Lipids are non-polar molecules, making them hydrophobic. Hydrophobic translates to water-fearing, and that means they are insoluble in water. Because of this characteristic, only the smallest lipids can dissolve into and travel through our bloodstream on their own. Larger lipids often combine with proteins, forming a lipoprotein. Lipoproteins are soluble because the proteins on the outside are soluble. Lipids can also dissolve into other lipids and organic solvents like alcohol. While more forms exist, we will briefly discuss the four most common forms of lipids: fatty acids, triglycerides, phospholipids, and steroids. Fatty acids are the first type of lipids we'll discuss. They are used to make two other types of lipids, triglycerides and phospholipids. They also function to make ATP. Make sure to know the difference between saturated and unsaturated fatty acids. Saturated fatty acids are saturated with hydrogen ions and therefore do not have double bonds in their structure. Unsaturated fatty acids have double bonds, so they are not completely saturated with hydrogen ions. The second lipid we'll cover are triglycerides. Triglycerides are the most abundant lipid in our bodies and in our diet. Triglycerides provide protection, insulation, and energy. They are the most highly concentrated form of chemical energy, providing more than twice as much energy per gram as carbohydrates or proteins. Our bodies have an unlimited capacity to store triglycerides in adipose tissue, or what's known as fat, on our bodies. Triglyceride structure is a glycerol backbone with 3 fatty acid chains. We've all heard of fats. Fats are triglycerides with mostly saturated fatty acid chains. These are solid at room temperature and tend to be less healthy for us. Oils are triglycerides with mainly unsaturated fatty acid chains. They are liquid at room temperature and are more healthy for us. For example, olive oil. The third lipid we'll cover are the phospholipids. Their structure is a glycerol backbone, 2 fatty acids, and a phosphate group. The phosphate group forms a polar head. The 2 fatty acids create the nonpolar tail. Because these molecules have both a polar part and a nonpolar part, they are considered amphipathic. The main function of phospholipids in our bodies is as the primary structure of cell membranes. The outer surface of each cell in our body is made up of a phospholipid bilayer, with the nonpolar tails of each layer facing each other as seen in the figure here. The last lipid we're covering are steroids. Their structure is very different from the other lipids we discussed. Steroids are flat molecules made of 4 hydrocarbon rings and sometimes a hydrocarbon tail, as in cholesterol. The ring area is nonpolar, so steroids are considered nonpolar. Cholesterol is an important and necessary steroid in our bodies. It gives stabilization to cell membranes and also is the precursor to other steroids like bile salts, vitamin D, and the steroid hormones. Bile salts are necessary for the breakdown and absorption of dietary lipids. Vitamin D is needed for the regulation of body calcium and for bone growth and repair. Steroid hormones include testosterone, estrogen, and cortisol, all important regulators in our bodies. The third group of organic molecules we'll discuss are proteins. Proteins perform a wide variety of functions. Proteins like collagen form the structural framework of our body parts. Hormones like insulin regulate body processes. Proteins like actin and myosin are responsible for the contraction of our muscles. Antibodies are proteins that protect us against foreign invaders. Hemoglobin is a protein that transports oxygen through the body. And lastly, enzymes are proteins that speed up reactions. Proteins are very large molecules made up of carbon, hydrogen, oxygen, and nitrogen, and sometimes sulfur. They consist of amino acid monomers attached to each other by peptide bonds. This creates a polypeptide. Polypeptides are then folded into complex 3-dimensional structures. There are 20 different amino acids, and the order of the amino acids and how the polypeptide is folded gives the protein its unique characteristics and function. If proteins are exposed to extreme environments like high heat, they denature, losing their unique shape, and they no longer function properly. Proteins have 4 levels of structural organization. The primary structure is a polypeptide made from a chain of amino acids connected in a particular order by peptide bonds. The secondary structure is when the polypeptide twists into an alpha helix or folds into a beta-pleated sheet. These are stabilized by hydrogen bonds. The tertiary structure is when the alpha helix or beta-pleated sheet folds onto itself and then is stabilized into that unique structure with more hydrogen bonds. The final quaternary structure is when 2 or more tertiary structures are bonded together. Enzymes are a type of protein that speed up reactions. Reactions can be 10 billion times faster due to enzymes. Enzymes are highly specific and only bind to certain cells. Enzymes are not changed or used up during the reaction, so they can continue to function in more chemical reactions. In this figure, the enzyme sucrase is in orange. Enzymes usually end in ASE. The substrate is what is being worked on. In this case, sucrose is a substrate. Sucrase, the enzyme, speeds up or catalyzes the hydrolysis reaction, resulting in glucose and fructose being built. The fourth and final group of organic molecules we'll discuss are the nucleic acids. Nucleic acids are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. DNA is a nucleic acid that forms a cell's genetic code and regulates its activities. RNA guides protein formation, and ATP is our body's energy currency. Each of these nucleic acids is a polymer made up of monomers called nucleotides. Each nucleotide has three parts: a phosphate group, a nitrogenous base, and a pentose or a 5-carbon sugar. Here we see the option each nucleotide has. First, they all must have a phosphate group, at least one. For the pentose sugars, it can be either deoxyribose, as in DNA, or ribose, which we find in RNA. Both DNA and RNA have cytosine, adenine, and guanine as their nitrogenous base choices. Additionally, DNA has thymine as one of its choices, and RNA has uracil as one of its. DNA, or deoxyribonucleic acid, is a double-stranded helix. Each strand is a polymer made of nucleotides. The backbone of each strand is made of the deoxyribose sugar and phosphate groups of each nucleotide. Sticking out from the backbone are the nitrogenous bases. The nitrogenous bases of each strand form hydrogen bonds between each other. Nitrogenous bases are specific in which nitrogenous base it will bind with. Adenine pairs with thymine in DNA, and in RNA, adenine and uracil bind together. In both DNA and RNA, guanine and cytosine pair together. You can see in this figure that the G and the C bind together, guanine and cytosine. And then you see A and T binding together, adenine and thymine. And this is DNA. Ribonucleic acid, or RNA, differs from DNA in its structure and function. DNA is found in the nucleus, whereas RNA is mainly found in the cytoplasm. DNA is the genetic material and the instructions for protein synthesis. RNA carries out the directions for making proteins. DNA is a double helix and RNA is a single strand that can be straight or folded. A very important nucleic acid in our bodies is ATP, or adenosine triphosphate. It has the components of a nucleotide. It has ribose as the 5-carbon sugar, the nitrogenous base adenine, and it actually has 3 phosphate groups, thus triphosphate. ATP is our body's energy currency. When something needs energy to occur, we use ATP to pay for it. ATP goes through a hydrolysis reaction with the enzyme ATPase. This causes one of the phosphate groups to break off, and that releases energy. And also, it ends up with an ADP, adenosine diphosphate molecule. Notice the di means 2, so now there are only 2 phosphate groups attached. The other one has been broken off and given us energy. In this lecture, we covered the four major organic molecules: carbohydrates, lipids, proteins, and nucleic acids. We also defined ATP and its role in the cell.